This lecture video is going to be on brain and cranial nerves. So again, let me get some PowerPoint. Okay, so this is the other half of the central nervous system. Remember, central nervous system or CNS has two parts, spinal cord and brain. So we already discussed the spinal cord and in detail. Hopefully you have watched that video over and over. So now we are going to talk about the brain. Okay, it is obviously one of the largest organ, the body. It's weighed like 3 pounds. You don't feel it that you have like 3 pounds right there and walking all day long on 3 pounds on your head. But there is some other reason. Like, why don't you feel that? Because it's like it's kind of floating in CSF, cerebrospinal fluid, which I'm going to mention later on. Okay, so brain controls your sensation. They have memory, emotion, decision making, behavior, all these things controlled by brain. This is the most upper and complicated level of your complete thought process, okay, or regulatory process, in other words. This is the outer structure. So in order to understand the brain really well, basically we have to first go into the embryonic development stage. I'm going there, but before we go, a couple of simple things that I'm going to mention here is, unlike spinal cord, where gray matter is inside and outside is like white matter, here the outside part is the white matter, meaning most cell bodies. That's why it appears in gray. And if you want to see the some white tracks, it should be inside the brain. So if you cut it open, if you make like a sagittal section of the brain, then only you can see that. Okay. So these ups and downs of the brain, those are this up region, this up area where my pointer is right now, is called gyrus. Gyrus, that's the word. Gyrus is singular, gyri is plural. Okay, and the groove, this groove or the shallow part, it's called sulcus. That is the word. Sulcus, or the plural will be sulci. This humongous part, which is like almost like 83% of the brain volume. So this humongous part is called cerebrum. That's the word, cerebrum. And this is cerebellum. This is cerebellum, the bottommost part. Right here, this is, we can call it brain stem. These are different part of the brain stem. You can see the entire brain stem starting from here if you open the brain. So if you go back to the previous picture, you can see brain stem up to here. So that's brain stem, commonly known as, okay? And this is cerebrum, this whole big thing, and this is cerebellum. Then the main part of the brain, like when you study the function of the brain, as well as structure, even different anatomy, you have to understand this is the cerebrum. Then this portion right here we call thalamus, and right below this is hypothalamus. Together, this area we call diencephalon. Dia means through, so diencephalon. So this is basically a relay center where all the information goes up to the brain and all the information comes through there. Okay. Then this region, like starting from here all the way down, we call them brain stem. Brain stem as a midbrain, middle portion of the brain, then the pons. That's you can remember in two ways. It's like a letter P right there, or pons means bridge, like this. Pons, it's a bridge. So that's the bridge between the lower part of the brain and the upper part of the brain. And this is medulla oblongata. Medulla means inside. So if this is the outside, all the way inside is this, the brain, and oblong means rectangular. So this is a medulla oblongata, means a rectangular inside area. And this is cerebellum, right, to be attached at the bottom of the brain, right there. It looks like a small piece of broccoli attached. That's the cerebellum. Okay. So now, how do we approach this chapter? The chapter you're going to approach, actually, you have two things to study. Okay, okay, three things to study. So basically, you need to know the anatomy very well. So when you go detail anatomy, that's your lab portion of the study. And now in this part, you are going to know the different parts of the brain and their function, as well as 12 pairs of cranial nerves. So 12 pairs of cranial nerve, meaning that's, remember, 31 pairs in case of spinal cord, here 12 pairs. So 12 pairs of cranial nerve, known as like CN1, cranial nerve 1, 1 Roman numeral, and CN2, cranial nerve 2, CN3, cranial nerve 3, and so on. But those have specific names, such as cranial nerve 1 is olfactory nerve, cranial nerve 2 is optic nerve, cranial nerve 3 is oculomotor nerve, cranial nerve 4 is trochlear nerve, cranial nerve 5, trigeminal nerve, cranial nerve 6, abducens nerve, cranial nerve 7, facial nerve, cranial nerve 8, vestibulocochlear nerve, cranial nerve 9, glossopharyngeal nerve, cranial nerve 10, vagus nerve, cranial nerve 11, accessory nerve, and cranial nerve 12 is hypoglossal nerve. So what I did, I put a separate document file saying all these cranial nerves, I just said that, I recited the names, all these cranial nerves, their name, their type, because nerve can be three types, we know. One is called motor nerve, one is called sensory nerve. So motor nerves bring out the information, sensory nerve bring in the information, and another is the mixed. Mixed means that nerve has both kind of tract. So some tract going in, that means sensory, some tract coming out means motor. Together we call mixed nerve. So nerve, those are the three types of nerve, sensory, motor, and mixed. So all these 12 pairs of nerves, some of them are sensory, some of them are motor, and some of them are mixed. So you need to know that. And then obviously their function and what happened if that get damaged. So for example, just a simple one. So olfactory nerve. So that is a sensory nerve because this brings in the information. It's right here on the nose. It brings in the information to the brain, which is known as cranial nerve 1. And so its main function is smell. So obviously, if it is damaged, you are not going to have smell. So that means you have impaired sense of smell. So that's how you have to memorize. So you have a smell, so impaired sense of smells when the olfactory nerve is not working, and so on. So what I did, I make a chart. I made a chart for you. The entire table is posted in the Blackboard as well. So if you whenever you get a chance, you download that and memorize that chart. That is extremely important, and that needs some writing and that needs some reading. So I would recommend you read that and then take a piece of paper and write that over and over at least 2 to 3 times, because not only you are going to need that for your lab practical, but also you're going to need lab quizzes, and but also you're going to need that for lecture exam 4. So in the lecture exam 4, you are going to get at least 3 to 4 minimum of 3 to 4 questions from that area only. So this, like as I said, it's a simple thing. I gave you the strategy. I make you the review note, and now it's up to you. So make sure you read that thoroughly and know exactly all the cranial 12 pairs of nerves, their function, their type in detail. Okay. And I'm going to mention of them a little bit in this lecture as well. Okay, so when you study the structure of the brain, the first thing that we should study is the embryonic development of the brain. So what does that mean? That means how the brain is formed. So initially, our nervous system, we begin with a neural tube like this. So like this. Let's say this is a neural tube. Now, that tube, if this neural tube has to grow, they will grow this way, they will grow this way, they will grow on the side. So it's like a tree. So when you planted a tiny one, like a tiny plant, and when it becomes a tree, it grows all the way. Like it grows on the top, it grows, root goes down, they become bigger. So this neural tube, if you look at this neural tube, then it becomes like this, my arm. So it becomes like big, tall. But here is the problem. The problem is when this is happening at the same time, the skull has formed, like our skull has formed. So then this neural tube, which want to grow, does not have room. So they are basically stuck right here. And it becomes, in my opinion, like a bonsai, meaning it does not grow this way, it becomes like this. And that mushroom head that you see, that's basically this. So that could not grow because your skull is very strong. So they become like a mushroom head. So this is the top part, so-called, of the brain that we're talking about. This is the bottom part of the brain. And so where is the middle part? Middle part you cannot see because it is kind of on top of this. So if this is the middle portion, the top part is covering this. So in order to see this, you have to remove this or you have to cut it. So if we cut it on the sideways, like a sagittal way, then this is the middle part that you can see. So you can see the top part, you can see the bottom part, but the middle part is kind of hidden. So when you study this embryonic development, so one more time, so the neural tube, now a couple of names, the rostral part. The word rostral means towards the nose, and word caudal or posterior means towards the tail. So basically what happened in normally we study the brain in like a rat or like a rodent model all the time. So that's how the name came, and we continue. But we can call this like a rostral side means towards this side, and the caudal side means towards that side. You can think that way. So the rostral side or the anterior side, they become prosencephalon. Prosencephalon, and then mesen means middle, so mesencephalon. So prosencephalon, forebrain, the top part, and mesencephalon is basically the middle portion of the brain, and rhomben, or the tail part. The rhombencephalon means tail part is basically the hind part of the brain, the bottom portion of the brain. So that's the top portion of the brain, that's a middle portion of the brain, that's the bottom portion. Now the top portion, when they're growing, they become telencephalon and diencephalon. So these prosencephalon now bifurcated into telen and diencephalon. The telencephalon eventually becomes cerebrum. So the telencephalon eventually become that cerebrum. So I have a brain model right here. So this is the part. So this part, this is the telencephalon. This is the telencephalon. This is becoming cerebrum. Okay. So now the diencephalon you can only see if you open that part. So if you open that part, this portion is like a diencephalon. The middle, right here, the middle portion, that is the diencephalon. So diencephalon are all these thalamus, hypothalamus, and epithalamus area. So this is like a thalamus area, this is hypothalamus area, and epithalamus is the top area. So these become the diencephalon. So the cerebrum become the telencephalon, become the cerebrum, diencephalon become the thalamus. Then this portion right here, detail we are going to learn later on, the anatomy. This portion right here, that's the mesencephalon, which become the brainstem or middle portion of the brain. And then comes the whole bottom part, like this whole section of the bottom part. So that basically meten and myelencephalon. So metencephalon become brainstem or the pons and the portion and the cerebellum. So this is the pons, that region is the pons, and this is cerebellum. This two, that's metencephalon, and myelencephalon, this portion which is become medulla oblongata and followed by spinal cord. Okay, so that is basically from the embryonic structure how we get to the full brain. It has to be in your head, otherwise you can't really follow this chapter. So the neural tube, when they're growing up, they have no room. Just remember, they become like a mushroom head. That is the top part. So that's basically this. That's just I just showed you. That's cerebrum. And then right underneath it is all the thalamus, hypothalamus, and epithalamus area. That's like a relay center mainly. Then comes the brain stem of the midbrain portion. Then becomes pons and cerebellum, and finally medulla oblongata. So now when you're approaching to read this chapter, you basically need to know cerebrum, diencephalon, brain stem, like midbrain portion, pons, cerebellum, and medulla oblongata, what they do, what is their function in detail. Okay, we're going to go there. But before that, this is basically exactly what I just said is right here. So this is how we are going to study them in detail. But before that, there are two other aspects of the brain. Number one, the brain has ventricles. Ventricle means chamber. And number two, about cerebrospinal fluid, the fluid that circulates not only brain, but also spinal cord. Now, brain, like the spinal cord, also covered by meninges. This is exactly the same way we learn in spinal cord. Meninges are same as spinal cord, meaning they are three layers: dura mater, arachnoid mater, and pia mater. So pia mater adheres close to the brain. Then next is arachnoid mater, and then outside is the strongest one, is like a dura mater. The same layer of meninges, they can be on the brain and the spinal cord. So this layer, if you imagine, this is your layer that goes surrounding the brain and then continue to the spinal cord. Okay, so same covering. Ventricles of the brain. The brain has 4 ventricles. The word ventricle means chamber. It has 4 chambers. Now, when you look at the cerebrum, one more time, I'll show you the model. When you look at the cerebrum, this is the humongous cerebrum. They have literally a huge fissure, a long groove that actually break this, that actually create the 2 parts of the cerebrum. We call them hemisphere. So this is one hemisphere right here. This is the right hemisphere, and the other half is the left hemisphere. So brain, the cerebrum, has 2 independent areas. This longitudinal fissure, which runs right in the middle, that actually divide the cerebrum into 2 independent hemispheres. Same thing here. So when you're looking at this cerebrum, this is one hemisphere, that one hemisphere. The reason I'm mentioning this, because one ventricle is right here, the other ventricle is right here. Instead of calling them 1 and 2 or 1 and 2, we just call them lateral ventricles. So lateral ventricle of a left hemisphere, lateral ventricle of a right hemisphere. So we have 2 lateral ventricles. Then right in the middle, we have a 3rd ventricle. Now, 3rd ventricle and lateral ventricles, they are connected. The 2 lateral ventricles, within them, they don't have connection. So they're connected through the 3rd ventricles. This tiny aperture, like a tiny hole, that's called interventricular foramen. Foramen means hole. In a bone chapter, we learned that. So interventricular foramen, that opening right there, so they connect lateral to the 3rd, they connect lateral to the 3rd again. Now then 3rd, so we have 3 already, so 2 lateral ventricles and 1/3. Then there is a pipeline that's called mesencephalic aqueduct. Mesen means middle, or you can call cerebral aqueduct or mesencephalic aqueduct, that connect this 3rd ventricle to the 4th ventricle. So 4th ventricle, which is actually at the base of cerebellum, that's our number 4 ventricle. So we have 2 lateral, 1/3, 1/4 ventricle, and then that continue to become central canal of the spinal cord. That's our 4 ventricles. So our 4 ventricles are 2 lateral ventricles, 1/3 ventricle, 1/4 ventricle. 2 lateral ventricles are not connected. They are connected through the 3rd ventricle using the opening called interventricular foramen. Plural will be interventricular foramina. And then this is mesencephalic aqueduct or cerebral aqueduct, which connects third and the fourth ventricle. Then fourth ventricle continue in the spinal cord, become the central canal of the spinal cord. So what is in the ventricle? Inside the ventricles, we have cerebrospinal fluid. That is commonly known as the abbreviated form is like CSF. Cerebrospinal fluid. So follow the name, cerebrospinal fluid, meaning that fluid is in the cerebral, which is our brain, as well as in the spinal cord, both. So they're approximately 80 to 150, 160 ml, but every day body is producing almost close to 500 ml of cerebrospinal fluid. So the fluid mainly comes from through the blood-brain barrier from the blood, and then we maintain the particular amount of glucose, proteins, and ionic balance, which is basically has a tremendous function for all the nerve cells and supporting cells. So what they do, they have three distinctive functionality: mechanical protection, chemical protection, and circulation. So obviously nutrients and waste products are going back and forth. Remember, we learned that astrocytes, they create the blood-brain barrier, so the blood directly has no contact with the nerve cells. This is the contact. Cerebrospinal fluid is the contact between them. So they get the nutrients delivered to the nerve cell, and they take the waste product back to the bloodstream. Then obviously chemical protection referred to that they create an ideal ionic environment so that the nerve cell can work best to produce and carry the action potentials or the signals between them. And finally, mechanical protection. Remember at the beginning, I said we have like 3 pounds weight of a brain, like a nerve tissue. You don't feel it. It feels like 50 grams. So with like instead of 1.5 kilogram, 3 pounds, it feels like 50 grams. Why? Because it is like floating in the CSF. It is floating in the CSF. For example, like if you want to pick up somebody in land, that person could feel like really heavy. But if you both are in a swimming pool or in a lake, and if you want to pick up that person in the swimming pool, you'll feel like very light. Same thing. It's called buoyancy. So basically, that cerebrospinal fluid, they're everywhere. So the brain is floating in them. So not only they are protecting the brain tissue, because all this nerve tissue otherwise is going to be damaged if you have a 3 pound sitting on top, particularly the lower portion, but also when there is a impact comes onto the skull, that softens down to this CSF, that softens down to the CSF, so the brain does not get that much mechanical damage. Okay, so cerebrospinal fluid has a lot of function. This is the summary: mechanical protection, chemical protection, and obviously circulation. Now, where they form and how they circulate, these are the two things we need to answer. So where they form in the ventricles. They form mainly in the ventricles. Why? Because in the ventricles, there are some modified ependymal cells. Ependymal cells, one of the supporting cells in the CNS. And these are modified because they create a network of colorful cells. That's why it's called choroid plexus. Plexus means network, choroid means colorful. So all the blood capillaries covered by the ependymal cells. And where are they? They are in the lateral ventricles, they are in the roof of third ventricle, they're in the fourth ventricle. So all the ventricles, they have these things, and that's why they're making lots of CSF. As I said, they can make as much as like 500 ml of CSF, but they're getting absorbed all the time. So almost 150, 160 ml circulating throughout your spinal cord and the brain all the time. Like, the question is how they circulate then. This is how they circulate. So I think this is the better slide. So this is how they circulate, meaning CSF formed in the lateral ventricles. We just learned they go through the interventricular foramina from the from the lateral ventricle. They come to the third ventricle. Third is in the middle, lateral is on the side in the two hemisphere. So they come in the third ventricle. Then from the third ventricle through the aqueduct, mesencephalic aqueduct or cerebral aqueduct, they go to the fourth ventricle. Now, fourth ventricle, from there is also two aperture, like two opening, like lateral and median apertures. I don't have a picture for it. It should be right there. So those aperture allow them to go to the subarachnoid space. And then subarachnoid space, they go around the brain and the spinal cord everywhere. And finally, they go into the venous blood. This part you are going to learn in ANP2. So venous blood, meaning on the vein, they collect the blood, they go take that blood to the heart, and then the heart send that blood to the lung, so that lung purify them. Purify meaning they get rid of the carbon dioxide, they put oxygen back into the blood, that blood come back to the heart again. That's why it says heart and lung. And then it goes to the artery, and when they pump into the artery, that artery branches into the brain, and they bring it to the choroid plexus again. So this part, obviously, the how vein, artery, heart, lung works, you don't need to know detail because that is ANP2 material. But right from here, you should know from all the ventricles plexuses, the CSF goes to the corresponding respective ventricle, and then that's how they travel. So CSF, what is CSF? What is their function and how they travel? These are the three things you know, as well as the ventricles of the brain. So we have four ventricles to start. Now, let me explain first how these slides, how I did the slide of the remaining one. Remaining one is I'm going to go through with you, but it is really a simple way I designed it. Right here. So I started from bottom of the brain to the top. So we are going to learn medulla oblongata, we are going to learn pons, then like a midbrain, and then thalamuses, and then the cerebra, this way, because the lower part of the brain, those are more autonomic function. Autonomic function, meaning you don't have any conscious control on them. And the more you go towards the up, more complicated functions like memory, emotion, music, language, all these complicated things comes right here. So we're going to start from the bottom. So this, the slide, when you see this kind of picture, that green area representing medulla oblongata, this is what we're going to study right now. So medulla oblongata, so this is a summary part. So as I said, this has all autonomic function. So basically, there are 5 cranial nerves, 5 of them. 5 cranial nerves, 12, 11, 10, 9, and 8. These 8 through 12, these 5 cranial nerves are in the medulla oblongata. And there, this area mainly, those are cranial nerves. We're going to talk about individual function a little bit later. But this is, they're mainly cardiovascular and respiratory center. The main, and also reflex center for coughing, sneezing, swallowing, etc. But this is really cardiovascular and respiratory. What does that mean? Meaning this area control your heartbeat. So that heart, when they're pumping, the blood goes to the artery and then they collect back to the vein. That pumping, as well as the diameter of the blood vessel, that control is right here, cardiovascular center. How they do it, do not need to know because that's ANP2. So cardiovascular center is located at medulla oblongata, meaning that is the area they control your heartbeat and obviously the movement of the blood through the blood vessels. They also have a rhythmic area for the respiratory center, like basic breathing, like inspiration, exhalation, inspiration, exhalation. That's basic breathing rhythm. That's here, but pons has a little bit extra work also on the respiratory center, and apart from these 5 cranial nerves. So now basically we are going to talk about those 5 cranial nerves. So this is right here in the medulla oblongata. Those 5 cranial nerves we are going to talk about, as I said, 8 through 12, all the like 12, 11, 10, 9, 8, those 5 nerves. So basically what you need to do, you can refer to that chart again that I I discussed with you earlier. That chart has the detail of cranial nerves. Go back there and study them again. Okay. So this is the last one called hypoglossal nerve, the number 12, hypoglossal nerve. Hypoglossal nerve, they are motor nerve. Hypoglossal, they are motor nerves, so they are mainly controlling. To control what? Control muscles for speech. So if your hypoglossal nerve cannot function properly, you will have an impaired speech. By the way, there are a lot of other nerve also involved here, but this is the main one.